So this is the little figures in our textbook of how PCR works, which is kind of true, right? So they're showing us, yeah, it's not really showing us anything. All right, so this is like the generic, right? We stick the primers on, each primer extends a strand, one goes one way, one goes the other. We do, you think by now I'd be able to do this? No, not going to happen. Right? We do a heat denature, we break the hydrogen bonds. As we cool it, the primers sit again, either on the newly made strand or back to the template. Make the new strands on both, heat it up, primer anneal, make the new strands, and so on and so forth. Okay. Yeah?
I was wondering, like, in the PCR tube, do you get water vapor where it's all happening?
Yes. So because of all that heating, so in the, which used to be a gigantic problem, so you would lose, right, if you're heating something to boiling, you're going to get evaporation of the water, or the aqueous part, which means everything gets more and more concentrated every time you heat it. And so in the good old days, you either had to overlay that water with oil so that it couldn't evaporate, but then you had to get all that oil crap out before you ran the gel, or it made like a mud slick in your electrophoresis and screwed up your band, so it was kind of a pain in the ass. Now, the machine that you put the tubes in have a heated lid. So the top of the tubes stay nice and hot, so it doesn't evaporate up. It just, if it evaporates up, then it comes, it condenses, comes right back down. So it keeps it from all jumping up into the top, so it really stays down in the bottom of the PCR tube. Because, yeah, if you're heating it 35 times, by the end it would all be up on the top, and there'd be nothing that's actually happening in your reaction. Yeah, so it keeps the whole thing almost the same temperature. Okay. Any other generic PCR questions for generic PCR? So when we talk about, yeah, go ahead.
Really cool.
Yes, that's good. We love PCR, right? We do PCR all the time, over and over in my lab. Okay, so regular PCR, when we just say PCR, we mean we are amplifying what? DNA. Okay? So that's our means to amplify DNA. Right? That's just regular old PCR. Let's think back to when I was blathering on in one of those lecture captures about cloning a gene into, I think we were talking about the cow, the bovine growth hormone, right? We're going to clone that gene into a bacterial plasmid, right? We used restrictions, we did the ligation, and we transformed the bacteria, and we selected them on the plate, and then we grew them up, and we grew up a ton of bacteria. We had that bacteria doing transcription and translation and making a shitload of that bovine growth hormone, and then we isolated that protein away from all the other bacterial goo, and now we have bovine growth hormone. Okay? If we did regular PCR from DNA for the bovine growth hormone gene, and we cloned the gene, so we got that gene from the DNA, and then we cloned the gene in the plasmid, and then we put it in bacteria, and then we asked the bacteria to do transcription and translation, what potential problem do we have? Introns. Pesky introns. Right? So what bacteria prokaryotes, what do they not do? They don't slice. Yeah, they don't RNA process. So if we really did that, why am I not getting a... What is going on, my friend? Oh. Won't give me a new slide. I'm looking for a little something to the slide forward. Is this an upgrade?
Yeah.
proliferated when I wasn't looking. Pesky child of mine. Like it's over there. Okay. So here we are. Oh, we're not there. We're here. Here's our plasmid. Right. This would be our PCR product if we just use primers and DNA, right? And it would have introns in there, but this plasmid, which really is double-stranded, so let's not forget that. Right, this guy is too. It's just blah. And then we put it back into, our good friend, the little bacteria guy, and they don't do RNA processing. We're going to make transcription is going to be fine, right? We're going to do transcription, and we're going to get a primary transcript. Let's just say it's 5' to 3'. And it's going to have the exons and introns. But then when we try to do translation, we're going to try to translate. We're going to start at the first AUG. But we're going to read right into that intron, which is random sequences, right? No codons, but the ribosome's going to make it have codons, sticking it in the A site, and it's likely it's going to hit a stop somewhere in an intron, right? Because it doesn't matter if you have UGA, UAA, all the stop codons in an intron because they don't end up in our RNA. But they would end up in this RNA if we did it this way. And so then we're not going to be translating the bovine growth hormone. We're going to translate whatever the first amino acids right after AUG, and then it's going to get likely to be truncated in the first intron. And even if it's not, it's going to add 100 amino acids that are not part of the real protein from that first intron. It's unlikely to fold up correctly and act like growth hormone. So we can't do that, right? So if we want to do this process, we can't just PCR out of DNA unless we did that and then we physically took, we could if we were like really ambitious. Right? If we could cut, right, if there just happened to be a good restriction enzyme right there, we could cut it and do our splicing ourselves in the lab and then get rid of this intron. But a lot of genes have hundreds of introns, and it would never be in frame, and it would just be a disaster, okay? Because we only want the exons to be in this plasmid because that's the only thing that bacteria can handle for translation, right? We want it to read all the way through just like an mRNA. But the bacteria are going to give us primary transcript with introns. It can't process to mRNA like we do, so it's totally screwed. So what we have to do is a different kind of PCR that involves reverse transcriptase. So the only reverse transcriptase activity that we've talked about so far has been in what process? Viruses, yes. And what other one thing did we talk about during replication at the ends of the chromosomes? Telomerase, right? Remember, telomerase was made of RNA and it could complementary base pair to the end. It provided that 3' OH to refill, but we could also add to the chromosome and actually make it longer, right? RNA producing some DNA is a reverse transcriptase activity. Okay, so reverse transcriptase can take RNA and make DNA. So the opposite of regular transcription, which is why we call it reverse transcription, because it's the reverse. Okay. Sorry. So the reverse transcriptase that we use in the laboratory is isolated from viruses, specifically from retroviruses. Okay, because a retrovirus is an RNA virus like HIV, there's a whole bunch of other ones, that their genetic information is RNA. When it sits on the surface of your cell, and remember we talked about those bacteriophage that poop their DNA inside, Well, this guy would poop its RNA inside, right, along with the enzyme, the reverse transcriptase enzyme that comes in the viral particle, and it will immediately reverse transcribe that RNA into what they call complementary DNA. Okay, so cDNA for short. So the C just is complementary. So again, complementary, complementary DNA. So it uses the RNA as a template to make DNA. Then it makes the second strand of complementary DNA. Now you have a double-stranded molecule that actually integrates into your very own chromosomes, and now we transcribe and translate all the viral genes for the virus. Yeah.
What if the transcription already occurred? What if splicing already occurred and the intron that was spliced, how are you going to do the transcription part of the RNA splice off?
Yeah, so you would want to do reverse transcription on a fully processed mRNA, not one that's a primary transcript, because we don't want those introns in there. So what would be a way that we could avoid getting that primary transcript? If we think about where those might be, yeah, don't go to the nucleus. Only use cytoplasmic RNA. So the kits or the processes that, the protocols for isolating RNA, if you just want mRNA, you want to break open the cells but not the nucleus. So it has to be a mild detergent that breaks open the cell, the nucleus is intact, you spin away the nucleus, it's still heavier than the other goo, get rid of all the nuclei, and then just isolate RNA from that cytoplasmic region. It should be all mRNA. Once it gets to the cytoplasm, it has to be mRNA. Yeah? Sure.
So when you're using the reverse transcription PCR, that's when you're using neutrality DNA to plug it into the plasmid?
That's right. That's if you want to clone a eukaryotic gene and be able to express it appropriately to make protein, because we don't want introns. So we'd have to go off of the mRNA to get a DNA to do the cloning. So regular PCR would be used if you want to clone the entire gene, including the introns, because you're interested in studying the gene or studying splicing, or you want to clone a regulatory region, or you just want to know, is that piece of DNA in this organism? Like I talked about making GMOs, right, a plant, and you do all these processes, but how do you know that your gene actually got into that plant? You did all of this, it's never 100% efficient. They grew, but what if it kicked out the plasmid when it was a little embryonic seed? And so you can take a leaf of all 10 plants you grew up, grind them up, and do PCR to see if your human DNA is in there. And then you could just do PCR off the DNA. And then you want to sequence it to be sure, or southern blot it to be sure it's your right sequence of human. And so then maybe out of those 10, 4 of them got the DNA and the other 6 didn't. And then you throw away the other 6 and you keep those and you collect the seeds and so on. So, yeah?
How can they possibly isolate one protein out of the bacteria so efficiently that it doesn't cause an immune response?
Oh, that's a great question. So isolating the proteins from the bacteria is actually not as difficult as it sounds. It's a little more complicated. So when you're making that protein, sometimes you'll add a little 4 amino acids to the end of the protein that is specifically targeted to something sticky or magnetic or reactive to an antibody. Not enough so it misfolds the growth hormone, right? But so that it's got a little tag on the protein. And so if it's something that an antibody recognizes, then that's pretty easy. Antibodies are extremely specific. They're only going to bind to that sequence. They're going to pull out only that protein from your goop, right? And then usually when you engineer those extras, you engineer a way to cleave it with some enzyme there, and then you pop off those 4 and the antibody, clean that away because the antibody's sticky. You can pull that stuff out. Now you have this nice purified protein. So yeah, you can't take just the slurry of bacteria and jam it in somebody and go, here's your insulin, and then they have an immune response to every bacterial protein, exactly. So you have to go through a bunch of purification steps to make sure you get there. Yeah. And? Yes.
So, RT-PCR is referring to reverse transcription?
Yes.
Oh, it's not real time?
No. So reverse transcriptase PCR has been around for 20 years, longer than that. Real-time PCR has been around for the last 10. Unfortunately, they named it the same thing as that acronym. Real-time PCR, for most people, is abbreviated as qPCR because it's quantitative PCR. Sorry. And so we call it real-time PCR, but we don't call it RT-PCR. So it's kind of confusing. Yeah. But I'll always refer to real-time PCR as qPCR, okay, for quantitative PCR. Or just write out the words real-time, right? I'll try to not confuse people. So, unfortunately, they picked a name that, and real-time is kind of a stupid name for it anyways. It's just quantitative PCR. It's not real-time. Okay. All right. I digressed. Yay! Recent advances in the field. Okay. So, this is going to walk through reverse transcription and then a little bit of PCR. And so I'm going to add a little bit here so you can add along with me, right? If this is an mRNA, that's always 5', here's 3', right? The 3' end always has a poly A tail. It's just shown here with 6 A's or whatever, but we know it can be hundreds and hundreds of base pairs long. Reverse transcription, just like a DNA polymerase needs a primer in order to start. Okay, so it needs a juicy 3' OH in order to start complementary base pairing. Okay, and so when it comes in with a virus, the virus provides those little pieces as well. So it just starts out ready to go with its own enzyme, and the virus does complementary DNA, the other strand integrates, takes over your cell, and then it kills it. Okay, so if we're trying to do this in the test tube, we'd isolate this mRNA, the orange guy, right, the cytoplasmic stuff, and it's going to have its cap, right? I don't really care about that. It's going to have its upside-down G, the phosphate-phosphate bond, the methylated guanine. But we have to provide a primer. If we want a specific RNA, one mRNA, we'd make the primer complementary to just that mRNA sequence. We'd make it, well, they just, like whatever, let's make this up. They didn't give us base pairs. It's like CCAGUUA, whatever. That's the template. That's part of the RNA. So if we just wanted that particular RNA and nothing else, that's all we want to amplify, we don't want to make a primer to the poly A tail, because those primers are going to sit on every single mRNA that are in your cell, and you're going to get reverse transcribed every single mRNA in your cell. Sometimes you want to do that, and so you use a primer on the poly A tail. Then you're going to get a sample of every single RNA that's being expressed. That would give you an expression profile of a specific cell. The expression profile of a nerve cell is going to be completely different. Well, not completely. They're going to have some of the same, right? All the polymerases and metabolism genes. So then they're going to have the neurotransmitter mRNAs compared to a white blood cell who's going to have all the polymerases and cell cycle and metabolism genes, but then it's going to have antibody and all that other stuff, right? So it gives you a profile of what genes are turned on in a cell. If you use this primer, that'll sit on the poly A tail and pick up every single mRNA. If you just want to get bovine growth hormone out of this skin biopsy from some poor cow that you chopped a little chunk of his ear off or something, right, then you'd want to make your primer complementary to something specific in that RNA, right? Make sure you get the whole thing, so you'd want to make it in the 3' UTR. You want to make sure you get the stop and all the codons and the start and the 5' UTR, right? So you don't want to make it somewhere in the middle, because then you're only going to get half of an RNA. If you clone that, you're going to make half of a protein, right? So then you'd make the primer complementary to that. And again, your primer is made out of DNA, deoxyribonucleotides, because those are cheaper and more stable. We could use RNA, because again, the polymerases don't give a crap if that 3' OH is stuck to a deoxy or a ribo. They're going to go. But RT adds DNA, right, on an RNA template. So it would, if we were making this, primer over here, we'd want to make it we'd want to make A, C, T, G, G, G. This is the 3' N, right? The RT would grab that 3' N and complementary base pair across until it falls off at the cap. It's not going to complementary base pair the cap because it's sitting in the wrong, it's on that phosphate, it's upside down. So it's going to get that first base pair in the 5' UTR. If you want all the mRNAs in the cell, then we'd make this one. It'll sit on everything, you put a ton of primers in, so there's enough in there, and there's tons of enzyme, and then it goes across. So you're making a piece of DNA from a piece of RNA. That's reverse transcription. Okay? And so that's all this is showing, right? If we did put this guy, right, it's complementary base paired all the way across. And then we would, at this point, once you have this RNA-DNA hybrid, right, because now this top part is RNA and this bottom part is DNA, the part of the reverse transcription enzyme, that complex that the virus makes, will essentially chop up that RNA off the DNA, and then it will go ahead and complement to DNA to DNA. So now we have two pieces, a double-stranded DNA. Okay. And so then from this point forward, we're essentially doing regular PCR. Because now we have double-stranded DNA, just like if we would have collected some genomic DNA, except that there's no introns in it, right? This was an RNA. And so if this was the first AUG, right, we had just, oops, what was that? To the stop UGA, 3' UTR, et cetera. Right? So that's all that's in this piece is just the exons, just the coding region, right? 5' UTR, 3' UTR, poly A tail, but no introns because they didn't exist in that original mRNA that was our template. So now we're sitting here with this piece of DNA. At this point, we do essentially PCR. Okay, so we have the DNA, we have to heat it up, right? They're showing us, ooh, look, hot stuff. Woo, sweaty. Okay, denature just means breaking hydrogen bonds. Two strands come apart. Anneal the primers. That means the primers are all in here while you're heating it, and the polymerase is in here, right? But they can't complementary base pair because it's so hot, they have to stay single-stranded, right? And our tack, I'll draw some tack is in here too, but it's not doing anything because it's hot. The DNA's naked, can't grab naked DNA and start complementary base pairing because tack is a DNA polymerase. So in that original tube that you were doing this in the lab, you had the mRNA in there, you had your primers to do the first strand and the second strand. That second strand of DNA needed a primer to start it too. So it would be complementary to that RNA on the 5' end. Right? The original one is on the 3' end, the next one's on the 5. We made that double-stranded DNA. We also have Taq polymerase in there. Okay? So it was just sitting around doing nothing while RT is doing its job. Now that it's double-stranded, we had all those primers in there, we had the Taq, everything was ready to go. Once there's no more RNA, reverse transcriptase just kind of hangs out, or we kill it on that next heat because we don't need it anymore. It was just for that very first step. The rest of this is all Taq polymerase, just like regular PCR. And so then when we cool it down, we get complementary base pairing of the primers to wherever they're complementary to. Okay. If we cooled it down to room temperature, the big strands would come together also. So because it's a little 20 base pair oligonucleotide, a bunch of nucleotides stuck together, they can complementary base pair at a higher temperature than the whole thing coming back together. Okay? Just because they're little. So they're going to stick on there, and then you raise the temperature back up to make sure we don't have the big pieces come together to whatever the optimum temperature for the polymerase to do polymerization. And so this Taq polymerase is 72 degrees. The ones that proofread, some of them are 68 degrees, but they're all within that range. So the big pieces aren't coming back together. Okay? But then this Taq polymerase binds, and now this would be the 3' ends, right? Has to be, and they're going to polymerize in that direction. Okay? And so they're showing us, ooh, look, it's going that way. This one's going that way. And then once it's extended, then we go back and heat the crap out of it to pull all those pieces apart. Okay? And this is just showing us, this was the original template, this guy. And this was the original template. And then these two guys in the different color are the new strands. So we can see this is where the primer was, right? It didn't copy the entire piece of DNA because it wasn't complementary up there. So that one still exists. This one, it didn't copy the entire piece of that one. Okay. And it really, if this wasn't the end here, it would just keep going. And if this one kept going, this one would keep going too. So it doesn't just stop where you want it to stop because it's just a polymerase doing complementary base pairing. It doesn't know what we want it to do. Okay. And then we go through the annealing of the primers again, and the primers can go back on their originals. This is where they went when that's all there was. Or, right, this one is now also complementary to this strand because this and this looks exactly the same because they're complementary to the same other strand. And this one are exactly the same sequence because they're complementary to the other template strand, or they are the template strand. And so now this primer, right, can go here. And now when it extends, it will stop at the end because that's the end of that primer. That's physically the end of that DNA. You can't complementary base pair to air. So now this piece will really just be this big. And this piece will start here and go to here. And so then what we get, okay, we're extending. Okay, we've extended, right? So it'll stop here. This one stops here. This one just keeps going, blah, blah, blah. And now we start over again. Now all the pieces can be templates. The original can be a template. All the primers anneal again. And pretty soon, right, as long as it goes on a new strand, it's only going to make these little short pieces. The ones that still go off the original template, yeah, are going to be long. This one's going to be long, but this one's going to be short. Okay? And so over 35 cycles of this, the majority of what's going to be left are just these small pieces. Right? As there's more of those there, probability of primer sitting on those rather than template, and on and on. So then, right, at each round, it enriches for just amplifying that little piece. Yeah, you're still going to get it to come off the original two molecules, and then, you know, the original two strands, but by 35 times, those are going to be in the minority, and everything else is going to be the exact sequence, including the primer, and everything in between. Just what you wanted to amplify. And so on, and so on, and so on, right? And so most of them, you know, are only this big, except for the few. See? Yes, yes? Oh my gosh, okay. Until you get most of them being the size you want, right? And then they'd show up on a gel as a single band, And then you either cut that out or purify it and then subclone that guy. And then that's just the DNA that corresponds to an mRNA. And we call that complementary DNA or cDNA. No introns. Okay? So then the bacteria can make the proteins just like we would. And that's how we can use them to do this for us and make a whole crap load of proteins. Okay. So real-time PCR, also known as quantitative PCR, is used to determine the exact copy number of either numbers of genes or pieces of DNA, and you can also use it as a reverse transcriptase. Reverse trans qPCR. Okay. And so that's usually why people use qPCR. Because if we do, let's say we want to determine if, okay, somebody has a cancer, somebody has a tumor, and we do a biopsy, and we want to classify this tumor and figure out what kind of chemo should this person get, what are the best drug targets, and so forth. So we need to know exactly what's going on in this tumor. We want to know how many different oncogenes is this one expressing, right? Because every tumor is different. Almost all of them have some dysregulated oncogene or some dysregulated tumor suppressor, and so we want to classify what its story is, okay? And we want to know how much is being expressed, because oncogenes really are just genes that control the cell cycle. So all of our cells have them expressed at normal levels. When you overexpress them, that's when they become a problem, right? If you overexpress more than one, your cell cycle's going and going and going, and then that's a bad thing, okay? And so if you just did RT-PCR, regular reverse transcription PCR, against those oncogenes, you're going to get a band on a gel. You're going to get something that's glowing on a gel, and you could say the difference between this cell has none, because you don't see anything, and this cell has a lot, because you see a nice fat, glowy band. Or this cell has medium, but you can't tell how much. You can't give it a number. You can't go 10-fold. You can't say 20-fold. And if the problem lies at the if you're 10-fold over normal, that's when we know this is a problem. But you can't tell from a gel whether it's 5-fold or 20-fold. Right? It's not quantitative. It's just qualitative. You could say all or nothing, but that's really it. With quantitative PCR, qPCR, you can tell the exact number of molecules of RNA of a specific gene that are in there. Okay? And so that's the power of this real-time PCR, quantitative PCR, or how many numbers of actual pieces of DNA. So if you're making a transgenic animal, right, copy number of the genes you've added can be really important in looking at the phenotype if you're trying to recapitulate some human disorder. You're trying to make a mouse model of multiple sclerosis or a mouse model of cystic fibrosis. You want to know, did it get one copy of the chloride channel gene, or did it get 10? That would make a huge difference. So you could use qPCR regular to look at the DNA and how many copies compared to a normal mouse who should have 0, right, that you could do with quantitative. You could say the mouse has that DNA or not by regular PCR, but you can't tell me it has 10 copies, 4 copies, 7 copies. And so that becomes really important sometimes. Sometimes we just want to know, is it there or not, and that's all we care about. We do cheap and easy PCR, or RT-PCR. But if we want to know numbers, then we have to do quantitative PCR. Okay? And so this is just a tutorial from a company. And so one of the first types of qPCR was called TACMan, real-time PCR with the TACMan probe. Copyrighted. I think it's Promega, actually. And so it has the same idea, but it's a fluorescently labeled probe, which is really the primer that sets it up. And so when we have our template DNA and we sit down this probe with our primer, as we read into it, it will start clipping away at this probe while our primer extends, right? Notice the 3' OH here, that our regular old polymerase Taq would start complementary base pairing. Okay, and so as it's adding nucleotides here, it's clipping off this probe. These are fluorescing. It lets off this fluorescent dye that was attached to it, and one single probe sits on one single template strand. So one molecule of probe is one molecule of template. So one unit of light is one molecule of your template. And then you do this, and it goes through a machine that reads the light, and it quantitates it. If there was 10 units of light coming off, that means there was 10 molecules of probe, which means there was 10 molecules of your template. So you can come up with an exact number of how many molecules of RNA were sitting there or DNA were sitting there in your sample. And then you always compare it to your controls and all that kind of stuff, right? Because it's one-to-one and it's molecule to molecule. And as long as you have some sort of dye that is that quantitative, which is the only reason it works, it's a one-to-one. Okay? And so that's quantitative PCR. It's all based on the same things, except it's displacing something that gives off light that a machine can read, and the computer can tell us how many copies are in there. Oops, sorry. And so what you read, which, what the hell is all that? It's just a computer printout. You don't run a gel and see nice little bands that glow. It's a computer printout telling you how much fluorescence came out and how many of each molecule you have there. Okay? So it's very quantitative. So if you really need to know exact numbers, then you have to do that. Okay? So it's just showing the same idea, right? So your probe is sitting here, and then your primer, and as you complementary base pair and then get that off, this guy gives off light, the machine counts the light, that's how many molecules are in there. So that's very exciting. I can see you're enthralled. Oh, and that's it for that. We have questions about that. So the qPCR stuff, what I really want you to know is that it's quantitative. Okay, and you can get exact numbers, and it's about giving off light, and it's a one-to-one, not total detail. Otherwise, it's the same thing. You have to have a primer, you have to have the polymerase, except this time the polymerase is displacing the probe that gives off light. Okay? And RT-PCR, we should know reverse transcription PCR, right? It's an RNA template, makes cDNA, and then second strand cDNA, and then it works just like PCR. If you want one RNA, you got to make it specific to that RNA. If you want all the RNAs in a cell, you can do it to the poly A tail and get everybody. And your primer on the other side is more complicated, and we're not going to talk about it. You have to add a thing, because you don't know every RNA sequence, blah, whatever. We can if we know where it came from. Yeah, it's much easier now because of the Human Genome Project. Yes.
If the virus jumps in
Good. Yes.
good to accurate, but doesn't jump to transcription, his reverse transcription he can't be replicated. But if he does bacteria make it into
You should, yeah. Yeah, or a eukaryote. Yeah, they have to send their RT with it because we don't have one. Except in polymerase, but it doesn't work on RNA. It just works on the, it just makes it off of the DNA. Okay, we're gonna keep going. We're gonna keep going. We're gonna cover DNA sequencing because up to now what we've talked about is what we see at the end is a blob on a gel, right? And if we're doing PCR and we know what our template is and we made our primers, we can calculate how big we're going to expect the band, right? If you make your primers 1,000 base pairs apart, you should end up with 1,000 base pair fragment if you've amplified it, right? And then you run gel electrophoresis to look for size, and then you run a size standard, what they call a ladder, of known sizes of DNA so you can compare them, right? So you run a ladder that's got 5,000, 4,000, 3, 2, 1, 750, 500, 250, 100. And then you see, you run that to load your wells next to each other, put on the electricity, run it down, right? The small guys go the fastest, the 1,000 should run it the same. So you can tell if the fragment you PCR'd is 1,000 base pairs if it's at the same level as that 1,000 in your size standard. But that's all you know about it. If you do a southern blot on that, right, and you take the complementary sequence to whatever you think you PCR'd and you label that with radioactivity or with a fluorescent dye, and then you take your gel and you transfer it to the membrane, make it stick there, and then you add the probe, it should only stick wherever you, if that's really what you think it is, it should stick to that 1,000 base pair piece, right? If it's not, it shouldn't stick there, and when you develop your film, it's blank. But you did a positive control, so you know your probe worked, and you know the assay worked, and you just know whatever you PCR'd isn't what you thought it was. And then you say, well, okay, I made these specific to this piece of DNA, but it's 1,000 base pairs out of the 8 gajillion billion base pairs in genomic DNA. Probability says those two could easily stick somewhere else. It's hard in probability for it to be the same size, but it's within the realm of possibility. So just because you get the right size doesn't mean it's the right thing. And at the end of southern blotting, if it's lit up, then it's your thing, okay? And then we might want to take that and clone it into our plasmids and do expression. And so once we clone it into our plasmid and it was a white colony and it has the right size insert, we want to make sure it's in the right direction and that no mistakes happen, right? Especially if we use Taq polymerase, which we know makes some mistakes because we're broke, we can't afford the proofreading kind. So we need to know the exact sequence in that plasmid, right? All the methods that we talked about so far, we can get a good idea that it's what we want and it's in the right orientation if we know where there's restriction enzymes that might cut, and we can do, but we don't know the exact sequence. We can't find a transition or a transversion that might have happened, right? A base substitution or a deletion of one base pair. 999 is going to look just like 1,000 on a gel. 900 is going to look pretty much like 1,000. Okay, so we want to be able to actually look at the nucleotides. Yeah.
If you treat your DNA with a bunch of enzymes that don't cut your 1,000 base pair sequence, but cut other places, wouldn't that pretty much eliminate the chance of PCR on the wrong thing?
No, if you cut up your genomic DNA, that allows it to not be all tied up, but all those pieces are still in your tube. It's less likely, but there's still going to be stretches of 10,000 base pairs where that restriction enzyme didn't cut, where your primers could have stacked. It is much less likely. It does help it that if it's the right size, it's almost always the right thing. But if you're basing your PhD degree on that being the right thing just by size, right? Or somebody hands you a plasmid and says, this is the expression vector for something that's essential to an experiment that's whether you graduate or not. Yeah, you cut it, yeah, it's the right size, but you don't spend a year working on it if you don't know what the sequence is, right? So you want to sequence it. You need to know that person didn't hand you empty vector or some other mislabeled crap they dug out of the freezer so that you're not wasting your time. So we want to know exactly what the bases are. And so there's a couple different ways that sequencing can be done. I'm not even going to talk about the chemical sequencing. That's so old. Of course, I've done it because I'm that old. But dideoxy sequencing is pretty much the way most sequencing is done if we're just talking about plasmid or looking at a PCR fragment. And it's based on DNA replication, complementary base pairing, using a primer that has to be anti-parallel, that you're adding to 3' ends, okay? And it's actually also based on size and gel electrophoresis. So it's kind of all the shit we've been talking about, again, together in another permutation where we can actually tell exactly where the polymerase stopped polymerizing and what base is on the end of that little fragment. Okay? And the way we do that is we have a whole bunch of DNA and we have a whole bunch of regular, why can't I just learn? I'll never learn. Regular DNTPs, regular nucleotides, deoxyribonucleotides to incorporate. We have a primer, and then it's going to do, the DNA polymerase can do complementary base pairing. Okay. But we also have in our tubes one of the A, G, C, or T that is F'd up. That's a bad word. And what do you mean by that? What is wrong with this picture? Looky here. Yeah, what do we have here? Juicy. Who loves that? DNA polymerase. Blasphemy! There's no juicy 3'OH. What if polymerase incorporated that? Can it add another one after it? No, there's nothing to grab onto. So it falls off and it's chain termination. Done. We're done with that piece. We know that little fragment ends in a T if that's the tube that only has all the normal nucleotides but bad T's. And then we have another tube with normal nucleotides, bad A's. Another tube, normal nucleotides, bad G's. Normal nucleotides, bad C's. So we know everything ending in this tube ends in a C. Everything in this one ends in a G. Everything in this one ends in an A. Everything in this one ends in a T. Because it has to. Okay? And then randomly, the polymerase, right, there's more of the good ones, right? So let's say this is the bad T. There's still some good T's in there. So it's just random where the bad one's going to get in. It might read AGCT, TTTG, CC, bad T! And so that's the piece. It's 10 nucleotides long. It ends in a bad T. On another molecule, the primer sat, and it went AGC, bad T! And that one's only 4. And then the next one got through the first T, but the third T is... And so randomly, every spot during this whole strand, you're going to, at least one time, every T is going to end up being a bad one. Right? So some of them are going to get all the way through till that last T. Oh, the bad one. So you're going to have fragments the size of every single spot there's a T. When you run gel electrophoresis, the small ones go to the bottom, and then they go by size all the way back up. You have a whole lane of T's, a whole lane of C's, a whole lane of G's, a whole lane of A's. And then each one is one nucleotide different size. So we have to use a special gel that's enormous and really, really thick so you can see the difference between one nucleotide, and then you just look and you can read the sequence knowing which lane is. If you fuck up the lanes and you can't remember which was C, you're done. Okay? So you have to know, ooh, I loaded the A's in this lane and the G's and the C's and so forth. Okay? And so that's the power of that guy. And so, right, chain termination is all about, if you have to have that 3'OH. So if we make these fake nucleotides, these chain terminators with no OH, it's going to, polymerase is going to fall off and that little piece is going to be done. Okay. And so the idea would be if you're sequencing your plasmid, right, your vector DNA is part of the plasmid, right, it's really circular, it's double-stranded. If you want to sequence this path, you would sit a primer up here, you're going to sequence in this direction. If you use a primer down there, then you can sequence on that direction. But for a sequencing reaction, we only use one at a time, not two. Right? If you want to PCR that, you'd add both. So you can use the same primer that you use to PCR to do sequencing, anything that's complementary, right, but you'd only use one of them because you want to just see along one strand, where are all the T's, where are all the G's, where are all the C's, by size that way. Okay, and so then this is just the picture of what I was yapping about, right? There's the template. Which one? What's the bad guy in here? The G. So these are the bad G's, right? The G with no juicy end. Look at him, how bad he is. Okay. And so then randomly, they're just showing us, in this case, the bad G happened to get incorporated the very first one. This fragment's going to be, this is the primer, 1, 2, 3, 4, 5, 6, 7, 8, 9 nucleotides long. In this case, right, a normal guy got stuck here, the bad guy here, the bad guy, right? And it represents all of the G's in that. And then another tube you do with the C's, another tube you do with the T's, another tube you do with the A's. You have all these different size pieces. Okay. And so then this is just our book's little representation of the same thing that we were just talking about. And so then when you load, right, all the ones from your A tube that terminated at the A's, right, in the well of your gel electrophoresis, so there's your gel, right, and then here's my, oh, that's terrible. There, that looks like that is supposed to be a lightning bolt. So we put electricity through the gel. Right, and remember DNA is negatively charged because all those phosphates, right, that phosphodiester bond has all that negative energy. And so it'll go towards the positive, right, if you put the small pieces towards the bottom. So these were the A's, C's, G terminations, and T terminations, right? And so if this would be the smallest one, so that would have been the first nucleotide, right? And that's a G. And so they say, ooh, look, there's the G, and then what's the next biggest is this A, ooh, and then, ooh, we have two T's, and then C, G, A, G, C, T, G, A. And that's how you read up that gel from the smallest, which had to be that first base pair after the primer, to the longest one at the end of your template. Okay, and so that's the same idea, right? This, they're just showing it sideways, right? So this would have been the top of our gel and the bottom of our gel. Okay, and so then, let's see, can I make it turn? No, it won't turn. Will it turn? No, he won't do it. Okay. Oh, everybody puke. Okay. Right, and so then we'd read, if they're telling us this is the bottom, this is the small, right, this would be GGA, GA, TC, AA, blah, blah, blah. And then they're just, so right in here, this is what the sequence is. So that's how DNA sequencing works. And now we actually I'm going to skip this and we're going to do this next time. Because now, instead of using, and so that required radioactivity. Because, right, those bands showing up, because there's so few molecules when you're doing this process, if you just stained with that ethidium bromide like we do with PCR and tried to look at it on UV light, there's just not enough in there for those bands to show up. So actually the primers are radio-labeled with radioactivity, and they're included in each one of the fragments, and so then that's how, then you run that gel, and then you put it up on film, and then the radioactivity gives off light wherever there's a band, and then you develop the film, and then that's what you're actually reading. And so that was the old-fashioned way. We don't need to do that anymore because now we can fluorescently label the actual chain-terminating nucleotide, and so then now it's the same idea. It's fluorescently labeled with a different color, and we send it through a gel. So it separates by size, but then it goes through a laser that reads the color that comes out. So it'll read T, C, or blue, green, yellow, red, and it's G, T, C, A, on and on and on. And then the computer just gives you a readout. So it looks like this coming through the machine, and then the computer gives you a readout that looks like that. And then the different colored waves of the different sequences. And so that's how we do it now. We don't have to run the whole gels and do all of that kind of stuff. And now we actually don't even do this. We just send it to a company to do it for us for $5. Because actually the machine to do this and all the reagents are more expensive than $5 a reaction. So it's not cost effective to do it yourself. And it's way easier to just send it to them. Yeah. Okay, that's all for today. We're going to be done 5 minutes early. What a bonus! What the hell is wrong with me? All right, guys, thank you.